This is TPP1, short for tripeptidyl peptidase 1. This little enzyme has big responsibilities in the body, and the lack of it can lead to big consequences. TPP1 is a lysosomal enzyme that helps break down proteins in the lysosomal lumen. Specifically, it cleaves the N-terminal tripeptides from a broad range of substrates. When functioning correctly, the proenzyme form of TPP1 is synthesized in the endoplasmic reticulum and makes its way through the secretory pathway to the Golgi complex. Here, the protein gets an important modification. Oligosaccharide groups, such as mannoses, get added to the protein during protein modification. An enzyme called N-acetylglucosamine phosphotransferase in the Golgi recognizes these proteins and transfers a phosphorylated N-acetylglucosamine group to carbon-6 of one or more mannose residues. Next, a phosphodiesterase removes the N-acetylglucosamine group, leaving the phosphate group behind on the mannose. This is considered a mannose 6-phosphate targeting sequence that helps deliver enzymes to the lysosome. M6P receptors in the Golgi membrane are responsible for recruiting enzymes with these targeting sequences and forming a vesicle to transport them to the endosome. At a higher pH in the Golgi, the receptor has a higher affinity for the M6P sequences. But at a lower pH in the late endosome, the receptor loses its affinity for the targeting sequence and the enzyme is released into the lumen. Additionally, the lower pH is responsible for activating the TPP1 proenzyme, also known as a zymogen, to its active proteolytic form. Once the late endosome becomes the lysosome, the TPP1 is able to degrade proteins using its catalytic triad active site to hydrolyze peptide bonds. 3 amino acids are brought together by protein folding and carry out ping-pong catalysis, involving binding a substrate, kicking off the N-terminal, binding water, and releasing the C-terminal end. However, if a mutation occurs in the gene that codes for TPP1, the enzyme may not be synthesized, it may not get delivered to the lysosome, or it may fold incorrectly, inhibiting its catalytic action. In patients with a TPP1 deficiency, proteins like ATP synthase subunits cannot get degraded once transported to the lysosome and begin to build up in the cell. When proteins accumulate in nerve cells, it can lead to progressive decline in motor function, vision, and language, as well as cause seizures. This is a genetic disease known as CLN2 disease that mainly affects children as they develop and can lead to premature death. CLN2 disease symptoms can be slowed with a drug recently approved by the FDA, known as Brineura, or generically as cerliponase alpha. Cerliponase alpha is a replacement enzyme for TPP1 that is administered to the patient in the form of TPP1's proenzyme. It is able to be taken into the cell via receptor-mediated endocytosis using M6P receptors that naturally reside on the cell's surface to reuptake any lysosomal enzymes that accidentally get sent out of the cell. Like the natural form of TPP1, cerliponase alpha has M16 targeting sequences that allow it to bind the receptor at higher pH and dissociate from it at a lower pH as it is transferred to the lysosome. Here, the enzyme is activated and it is able to cleave peptides to prevent further buildup. After a little while, the replacement enzyme is degraded and the patient must be administered another dose of Brineura. While it does require continuous treatment and it is not a cure, Brineura has helped many families see a slow progression in their children's symptoms and likely extended their lifespan.